It's my great pleasure to introduce Professor Eric Seabloom here. And Eric is the interim director of the Cedar Creek Long-Term Ecological Research Observatory, which is a facility that has a really long history at the University of Minnesota. In that long history, there have been groundbreaking experiments helping us to understand ecology, resilience, ecosystem interactions, and surprisingly few pieces of work understanding the Earth surface processes around the creek and the landscape in general. And that's something that he and I were discussing as ways to perhaps bring the SAFL community and the ecology community together. So this is going to be a different style of talk rather than a cutting-edge fluid dynamics talk. This is a big picture possibilities in our neighborhood kind of talk. So thank you, Eric, for so much for being here.
Yeah. All right. Thank you so much for the invitation, Andy, and I'm really excited to be talking to you all. So there's going to be, already had some great conversations, which have been super fun, but it's going to be kind of a few different themes woven in here. So one, like Andy said, I want to introduce you a little bit to Cedar Creek, which is one of the field station being run by College of Biological Sciences. It's just 45 minutes away up in the north. Also, there's a lot of, like Andy said, there's a lot of long-term research here. So there's kind of a underlying theme of what makes long-term research different than other kinds of research. And then there'd be kind of like an embedded piece in the middle. Most of what I work on is terrestrial community and ecosystem ecology. So there's going to be a little bit of that in there. And then I'll come back and talk a little bit about some of the opportunities for work up at Cedar Creek. And hopefully, I mean, this all came out of hoping to kind of drum up some interest to do a spring tour when the weather is nice. We have lovely Cedar Creek here and some great systems. So if you're interested afterwards, get in touch and we can organize some kind of tour, which I think would be super fun. There's also going to be a symposium over on the St. Paul campus on March 23rd in the morning. So if you're interested in learning more about Cedar Creek, seeing a sampling of some different studies being done out there. So Cedar Creek is a research station run by the College of Biological Sciences. It's about 45 minutes north. It's right on the edge of Isanti and Anoka County. And it's a real interesting mosaic. So it's about a third of the landscape. You know, if you look out over here, you can see all these open areas. These are abandoned agricultural fields. There's boreal forests, deciduous forests, and wetlands. So there's these really nice creek and wetland systems. We've got 4 lakes of different sizes that range from Fish Lake, which is about 150 hectares, down to tiny little Ice Lake, which is, I don't know, twice the size of this room or something, more of a pond. We have deciduous and boreal forest, oak savanna that we maintain through a big long-term fire program, and lots of prairie and retired agricultural land across the landscape. So you can see some of the diversity here by looking at this vegetation map. So all the different colors are different vegetation types. And this has led to a real just diversity of animals associated with it. We also have 4 lakes, like I mentioned, of different sizes. We have over 2,000 species of insects on site. 75% of Minnesota mammals have been recorded at Cedar Creek, and 56% of the bird species that occur in Minnesota have been seen at Cedar Creek. So this has led Cedar Creek to be recognized as an area of outstanding biodiversity significance by the Minnesota Biological Survey, and it's a national landmark through the National Park Service. So I'm going to do kind of a tour, a quick 90-year tour by the sort of multiple decades of some of the work that's been done there at Cedar Creek and how that work continues, and then focus in on sort of a subset of it. So Cedar Creek was first formed in 1942, but work there actually started quite a bit before then and was really focused on aquatic work. So this is Raymond and his wife Eleanor Lindeman. So they were working in Cedar Bog Lake in the 1930s. This led to a publication called Trophic Dynamic Aspects of Ecology, which is really one of the first times that someone tried to parameterize a carbon energy-based food web, and really led to a lot of what we think about as ecosystem ecology today. I love this photo, a little slide of them out sampling out there in the marsh. And we have a lot of work that's continuing today. So Jim Kotner is in our department. So he is an aquatic limnologist mostly, and he's got an instrumented buoy out in Cedar Bog Lake, and he's looking at carbon fluxes and methane fluxes. And then as Andy talked a little bit about, Chris Leonard has been leading a restoration of Cedar Creek. So the lower part of the creek is still very meandery. The upper part was ditched in the 1900s. And so next winter, this coming winter, a year from now, they're going to be re-meandering that creek. And Andy had a great freshman seminar where the students put together an amazing plan. Like, I was like, wow, they should start their own company, like, consulting company. Did an amazing job. Sort of starting in the 1960s, there was also a real push in wildlife biology. So this was a really interesting time right after after World War II. There was a lot of technological development, and then the 1950s and 60s, there was application of this miniaturization, especially of radios. And this was one of the first places where people did radio tracking of animals in the wild, which obviously opened up huge insights into how animal behavior works in the wild. Love these photos, a cool old van there, and got the extension cords out doing radio tracking. They had some amazing systems where they had this big wall of lights that they would light up as radio signals were picked up across the reserve. And this work continues today. So there's a lot of different wildlife projects going on. A couple I wanted to just put up because they also have kind of fun websites you can look at where we do citizen science classification and identification of animals. So one is the red-headed woodpecker project. So this is Elena West up there with one of these lovely red-headed woodpeckers. So these are species that are rare and declining throughout the state. They're really dependent on oak savanna. And she leads a largely volunteer-driven program monitoring all these. And this red-headed woodpecker cavity camera is online, and they have cameras pointing in, and they monitor the nest success. And then we've got a grid of 100 motion sensor cameras across the whole 5,000 acres. And we have another website called Eyes on the Wild. These are just a kind of sampling of some of the photos that show up, but you can log in and kind of screen through and look at, 50 photos of waving grass, and then maybe you get lucky and see like a black bear or a fisher or something pretty cool. And then this was a time when there was a lot of collaboration with the forestry department, looking at interaction in terms of fire ecology and savanna restoration. And so the whole southern half of the reserve is divided up into large units, burn units, maybe 40, 50 hectares each and larger. And these have been applied experimental treatments of different burn frequencies. This experiment's been running since 1964 continuously. And this work has really expanded to include a lot of these grassland systems where we have large grassland burns where we divided fields in half, burn one half and the other. We also have reintroduced bison experimentally and looking at interaction between savanna restoration and these native herbivores. The 1980s was the start of the long-term ecological research site. And this is a time when we really had a big push into a lot of terrestrial, community, and ecosystem ecology. The long-term ecological research program is a program at National Science Foundation that was started in 1980. And it's a big grant, so the reserve is really the property. And this is a big grant that funds some of our signature research. We've got a really big prairie diversity experiment here. These kind of weird looking rings are a carbon enrichment project that's been running since 1988. So open air carbon enrichment. But I want to talk just a little bit about this LTR program, which is really pretty amazing. So in the late 1970s, this came out of the Biosphere Initiative. People were realizing how slow ecosystems were to change, and that there was a real problem because funding cycles are obviously much, much slower than any sort of ecosystem change that can occur, and even much slower than that ecosystems are changing slower than whole careers of people. And there needed to be a way to kind of bridge this gap between how people were studying things and the rates at which ecosystems were turning over. And so they started this LTR program in 1980 with an initial call to fund sites on a 6-year renewal. And so the first 4 sites were funded in 1980. Cedar Creek was part of the next round of sites, so it was funded in 1982 and has been funded continuously. University of Minnesota is very lucky because we also have the newest LTR site, so the MSP LTR, which you can, I guess, see out the windows, right? So this is an urban LTR, which was funded in 2020? 21? Okay, yeah. So now there's about 28 LTR sites, and they're located mostly in the US, but also some in Antarctica and in the, on Moorea in the Indian Ocean. And this is a really wide range of sites. So it includes rainforest. There's 2 urban sites, so MSP and Phoenix. There's sites in the Arctic Ocean, in the Antarctic Ocean. There's kelp forests and coral and open pelagic systems off the coast of California. And so these are really, these are really disparate sites. So there's kind of always a little bit of a tension of how do you sort of have a network when everything's so different. And one of the goals of these is not so much like, there's these big observational networks where you're collecting exactly the same data everywhere. These are very system focused. We're looking at mechanistic understanding of really important ecosystems around the world. There are some underlying principles that tie them together. We all carry in our wallets, if we work for the LTR, this list of the 5, or I guess 6, right? You have the social scientist part, right? So all of the sites are focused on monitoring primary production. There's population studies of the most important organisms at the site, looking at fluxes of organic matter and organic matter, inorganic matter, and also disturbance patterns. But how these happen is obviously quite different. You measure primary production in a very different way if you're in the California coastal current versus in a wetland in Georgia or in an urban environment. So I'm going to kind of dial in the center part of this to kind of build out some of the work that's been done at Cedar Creek and to really, I hope, kind of highlight how long-term research is a little bit different than other kinds of research, and not just because it's long, which is sort of the obvious part of why it's different. And so I'm going to talk a little bit about sort of long-term collaborations. And so what makes science interesting isn't just the data, right? There's a lot of data, and there's a lot of, it's really not all long-term data or not all data are really that interesting unless someone interesting does something with it, right? And also to talk about answering questions versus collecting data. So there's a difference in focus on, are you a data generation organization or something focused on answering questions? Then I'm going to move into talking about how experiments play into this. So what do we learn from experiments that's different than observational studies? And then thinking about, this is a site-specific work mostly, but how do we test generality through leveraging large-scale collaborations and global networks? So I wanted to start talking, something when I first got involved in the LTR, I didn't really realize is how important the people interactions are. And I'm sure you see that here at SAFL, that when people work together for a long time in a cross-disciplinary way, people start to change, right? People come to these seminars, maybe some of the seminars, you're like, I don't know what this person's talking about. But it starts seeping in, you interact with people, and people become more interesting. They become more interdisciplinary, their questions are broader. And I think this really plays out over long periods of time, and it's made a huge difference at these different LTR sites and other places, kind of like here, where people get kind of put together in close proximity and are just interacting and learning. And Cedar Creek is very much that way, so we have a core leadership team of about 17 people who oversee the main grant. These are people that represent a really wide range of different disciplinary backgrounds, but also different timescales that they've been involved. So we have people who've been involved since the very beginning of the site, but every year the team has stayed about the same size over the past 4 decades. But there's always a couple people who sort of roll off and move on to other things. Some new people come in. So we have a real mix of people who are just getting started at the site, people who have been around a long time. And this mix is really kind of drives a lot of the exciting science that comes out of Cedar Creek. We also have really long-term staff, and this is really also critical, and we work really closely together. So the staff are keeping the experiments going and putting in new experiments, maintaining the treatments. I mean, I was getting a great tour down in the basement, and I'm sure you have the same thing here. It's just like all the people are down there actually building things and putting things together. And then we have a lot of students and postdocs working at the site. So in the past 4 years, we've supported about 12 postdocs, 15 graduate students, and every summer we have about between 40 and 60 undergraduate interns working up all summer. And so we're doing lots and lots of mentoring of early career scientists. And so all of these different people working together has made Cedar Creek really, really productive and impactful scientifically. So in the past, I'm just looking because I have a few numbers here. In the past 4 years, Cedar Creek has published over 200 papers in 64 journals, so 34 papers in Nature, Science, and PNAS. And I kind of went back on like Google Scholar, and I was looking at citations over the past 5 years. So there's about a Cedar Creek paper, there's about 100 Cedar Creek papers every single day that get cited, if you look at the citation rates. So these are very impactful papers, and I think that happens because of the people working together. It's not just that it's like this amazing, there's a lot of cool data sets, but it's people really pushing forward in interdisciplinary ways to not just generate a lot of papers, but papers that are changing the field. We also have a big impact on the larger scientific community, so all of our data are freely available. So we just post them all. And about a quarter of all the papers that use data from the Cedar Creek LTR are people that have sort of no formal affiliation with Cedar Creek whatsoever. So the people who've just downloaded the papers or downloaded the data and written a paper from it, which is really pretty cool. Like that's kind of what it should be. So I'm going to kind of hone in on kind of just a 40-year little journey through a particular set of projects, so maybe you can get a vision of how some of these things evolve at this site. And this is going to focus on sort of human impacts on ecosystems. So this is a big focus of a lot of the work we do at Cedar Creek. I don't probably need to say this in this group, but humans are having a lot of different impacts on the environment, right? So we're burning fossil fuels, that's releasing carbon and nitrogen into the atmosphere, changing fluxes of other elements like phosphorus. These things then feed back on the climate in complicated ways. The changing climate can then interact and change disturbance regimes, like increasing pests, increasing pathogens, changing fire frequency, which feeds back again. I found this photo on the NASA website. So this is from the summer of 2022. So this is Oregon and Washington. Here's the Columbia River. And this is just a few of the late summer wildfires occurring in 2022 with the smoke plume going out over the Pacific Ocean. You know, this is going to change the climate as well as releasing carbon and nitrogen back up into the atmosphere. And it can be really, so understanding human impacts can be really tricky because so many things are changing at the same time and all feeding back on each other. And this can also be a challenge because we're creating combinations of things that haven't occurred in the past. And so what I want you to think a little bit, there's a lot of really great observational networks. So for example, monitoring CO2 fluxes, like with this eddy covariance tower. And those observational data are really, really powerful. But they have some limitations because making inference from observational data, it relies on the future being sort of similar to the past, right? But as we're moving forward into a future where we don't have historical analogs, we're going to be facing combinations of things that are going to have different covariances. So we're going to have different combinations of nitrogen, of rainfall, of temperature, different species being present. And so that can cause a limitation on our ability to make a forecast into the future because we don't have good analogs for it in the past, which means that more of a mechanistic understanding is really important. So setting up models or experiments where we're breaking apart some of the things that covary in the past in specific ways so we can really understand causal interactions. So as a really simple example, here's a figure showing CO2 levels in the gray line from early or mid 1800s up through the present day. So it's gone from 280 parts per million to 420. At the same time, temperature is changing, right? So these are temperature anomalies. All the blue lines are temperatures below the long-term mean and the height, and these are above. So we're getting both increased CO2 and increased temperature at the same time. And both of those can have really independent and ecosystem effects that work through different mechanisms. So one of the things that Cedar Creek is and with these LTRs, it's really focused not just on collecting data, but on sort of answering and understanding questions, which is related, like you need data to answer questions, but it also means that some of the studies there are evolving and building up over time. So there's some initial studies, there's things that emerge, there's new studies that are layered on top. And so what I want to do is kind of sort of show you a little thread of one of those examples. And this is focused on some of the abandoned agricultural fields. So this is an aerial photo of Cedar Creek. You can see all these open areas, which are now prairies. These are all retired agricultural lands. So Cedar Creek is on the Anoka Sand Plain. So it's basically just, it really is like a sandbox up there. The agricultural lands are pretty poor. So a lot of these fields were farmed for a little while and then taken out of production. And we've been monitoring those. We set up a monitoring program in 1982 when the LTR first started, and we've been monitoring them ever since. But we also know when each of those fields was taken out of production, so we know how old it was. So we have a chronosequence of land abandonment from fields that are just recently plowed all the way up to fields that haven't been plowed for almost a century. And this work was initially started to really understand sort of basic ecological principles of succession and how ecosystems and community change, but it's become much more relevant now as with increasing CO2, people are realizing that these abandoned agricultural lands and grasslands can store a huge amount of carbon. So about a third of the Earth's terrestrial carbon is stored in grassland soils. I read a study recently, it said that they back calculated that the plowing of the Great Plains was probably the largest human-caused release of CO2 that they've found, which is, I mean, it's not like dramatic like a fire, right? But then you're like, oh, wow, that's incredible. So we've been monitoring these fields. So here's a graph. On the x-axis is the years since agricultural abandonment. We have about 24 fields, which are the different colors. And then we've been monitoring them through time for 40 years. And so the jaggedy lines are basically the annual variation through time. And so this is showing plant biodiversity. And then this is all relativized to an unplowed sort of reference field that we have on site that's never been plowed, like I just said. So you can see across this century that we kind of recreate, plant biodiversity is continuing to increase, but hasn't recovered fully even over the course of a century. And also that plant productivity, so this is measured as above ground plant biomass, is also increasing, slowly going up, but with lots of variability happening. So this question, like I said, is also tied into soil carbon storage. And this is one, like in 1980, people weren't really thinking about, we should really be thinking about how much excess CO2 can get sucked out of the air and stored in old ag fields. But all of a sudden, that's a more relevant question. So here's a paper taking a look at some of that. So this is same set of fields, years since agricultural abandonment, and this is cumulative soil carbon stored in the top meter of the soils. And so you can see over time, if you take these marginal lands, which actually are not really economically viable for the farmers anyways, if you take them out, they'll continue to accumulate carbon just on their own for at least a century. So going back now to introduce some ideas of experimentation. So at the same time, so that we're observing that, but there's a lot of other things happening, right? So there's increasing temperature. You know, we burn fossil fuels, and mostly we talk about CO2, but plants are also made up of nitrogen. So there's a lot of nitrogen released, which comes back down as nitrogen deposition. So some of that carbon accumulation could also be confounded with increasing nitrogen deposition, for example. And this leads to thinking about the need for experiments. And long-term experiments are really costly and kind of challenging to do. And so an important question is, do we really need to do long-term experiments, or are long-term can you predict long-term outcomes from a short-term experiment? So this is just kind of a conceptual graph where you can think about sort of years of a treatment being in place, and this is the treatment effect. Woo. I'm going to stay away from the screen. I think I got to stay away from that speaker. So the gray line is showing what would happen if a short-term experiment was all you needed, right? So you have some treatment effect, so whether it's adding nitrogen or putting in a drought or burning it, and that effect equilibrates pretty quickly and then stays constant. But you can also imagine that there'd be positive feedbacks, or you might get stronger and stronger effects through time. You could have negative feedbacks, where you might get an initial really big effect that could fade away if the processes are attenuating. You can also see perhaps lag effects, where you might do an experiment for 15 or 20 years and nothing happens, and then all of a sudden you start seeing a big effect. Or it could be, some of the signal could be masked with external forcing. So think about those graphs, with all the jaggedy lines of those fields through time. It might take a long time to just pick out a signal against this background. And all of these, we see all of these, except we never see this, seemingly. So really what we found over 40 years of doing experiments is rarely do short-term experiments predict long-term outcomes. So I want to have you kind of think about that sort of general idea in the back of your mind, as well as maybe learning a little bit about grasslands and carbon storage. So one of the first experiments, or the first experiment we set up at Cedar Creek, were in these old fields. And so we took individual old fields, divided them in half. Half of the field was plowed. The other half was left unplowed. And then these smaller plots, which are 4 by 4 meters, were subjected to different nutrient levels, mostly a nitrogen gradient. And we've been doing that same experiment and monitoring soils and plant production and a lot of other things for the intervening time. So I want to just show you some of the outcomes of that experiment. Maybe it's interesting in its own right, or if you don't really care about grasslands and carbon, maybe it's interesting from looking at how treatments change over long time periods and thinking about that. So here's a figure showing above ground biomass with just 4 treatments. So tilled or not tilled, so it's just tilled once at the beginning and recovering, and then with and without fertilization. So the dark blue line is fertilized, and the dotted line is tilled. And so if we just look at these control plots, this is across the first 25 years of the experiment. You can see when you till the plots initially, you get a big increase in biomass as you aerate the soils, release a lot of nitrogen, the microbes are growing and nitrifying, but that effect fades over time, over about a decade. So that's one of those cases of sort of attenuation. That's not true when you fertilize. If you fertilize, you can see this tilling effect persists for 25 years. So this is very different just with these 2 treatments. And if you look at the effect of fertilization, so just comparing the gold and blue lines, there's a huge effect of fertilization for the first few years. It attenuates, and then it becomes much stronger again over time. So just in this one graph, there's kind of like all the possible ways you can start to see these experimental effects changing over time. It's the same thing in the community. So this is looking at plant species evenness. So this is how evenly distributed the abundances are. So even community, if you had 5 species, it would mean every plant had 20% of the biomass. So you can see that over time, just in the field, evenness is increasing. So plants are becoming more evenly distributed. When you fertilize, we're actually getting much more even communities for about 15 years, and then that effect disappears, which is kind of crazy. And then the tilling effect, you can see there's no tilling effect on evenness going out to 20 years. And then all of a sudden, the tilled plots, 20 years after they were tilled, become much more even. And this occurs because of the recolonization by nitrogen-fixing legumes. And they're just really bad dispersers, so it takes them 15 or 20 years to start to show up. So again, just in those 2 graphs, we've seen all the possible ways of treatments getting stronger, treatments getting weaker, treatments switching directions. So I want to dial into the soil carbon piece then. So this is looking at just the tilled plot. So this is sort of mimicking an agricultural field. So here's 40 years of data of soil carbon after a field was tilled. The dark line with the dashes is unplowed, or I mean it's unfertilized. So you can see that for 40 years, we get a nice slow increase in soil carbon just from stopping the tilling. And then these colors represent increasing amounts of nitrogen. And so as you add more and more nitrogen, you're increasing the soil carbon with these high fertilized plots. So these are up at agricultural levels. You're accumulating carbon about 3 times faster than in the control plots. This isn't making the argument that we should go out and fertilize fields. Like that actually doesn't add up because producing ammonium nitrate takes a huge amount of energy. So just to emphasize that we can't fertilize our way out of the carbon issue. But in a way, you can think about this as sort of a ancillary benefit that sort of mediated some of the effects. So there's a lot of negative effects of excess nitrogen. So you get invasive species, it can get in the groundwater and cause problems with aquatic ecosystems. It can affect loss of native plant diversity, increased pathogen loading. But in a way, we have had some, there has been a little bit of a sort of side benefit of this increasing carbon. But because of all the other effects of negative effects of nitrogen, which cause a huge cost to our society and health and ecosystems, there's a lot of interest in regulating nitrogen deposition. And nitrogen deposition's been going down in many places. I was on sabbatical a couple of years ago in the Netherlands, which if you look at a map of nitrogen deposition around the world, it's like the big black blob of like the highest. It's amazing. And so I got thinking about, so what happens if you stop fertilizing, like we don't have all this nitrogen, is all this excess carbon then just gonna like blow out into the atmosphere? And we could actually look at this. So in this experiment, some of the plots, half of the plots were fertilized continuously, which I showed you before, but half of the plots were fertilized for 10 years, and then we stopped fertilizing them. And so if you look at soil carbon here, so this is during the fertilization period. So in 1985, they've been fertilized for 3 years. These are the plots that are going to be fertilized continuously. These will have the cessation, and the blue are unfertilized up to high fertilized. So you can see that fertilization effect on soil carbon even in the first 3 years. And then this is just looking at the time frame from 2010 to 2025, so the last 15 years of the experiment. These are continuously fertilized. You can see the fertilized plots have much higher soil carbon. So this is kind of the flat part of that asymptotic curve. These are the same, these were fertilized only for 10 years. So they're a little bit lower, but you can still see that fertilization effect. So these have not been fertilized for 30 years. So the soil carbon that's stored in there is stored in a form that's robust and hasn't disappeared and is being held at least for 30 years as long as the soil isn't being tilled. So that's kind of good news that we didn't just like lose it all by having lower levels of nitrogen. Like I said, there's a lot of other things going on in these fields due to nitrogen. So for example, here's plant species richness. And you can see that as you fertilize, you lose more and more species. So you go from maybe 8 to 12 species down to 4 species, most of which are exotic perennial grasses. And so there's this interaction. So these fertilized plots are more productive, but also lower diversity. But there's a lot of theory that suggests that diversity should increase plant production. And so this motivated another experiment. Here we're causing diversity to decline by adding nutrients that's causing production to go up. So you would think this would lead you to this sort of negative relationship, like the most productive plots are also the lowest diversity. But that's kind of at odds, so you might see that in the natural world, but that's muddling up some of the mechanisms. So this motivated a really big experiment, which Dave Tillman put in 1994. So these plots are 9 by 9 meters. There's about 320 of them. They're put out and planted into randomly assembled mixtures of native prairie grasses, ranging from bare plots, so 0, all the way up to 32 species, and then we maintain these through weeding every year, and then measure soil properties, plant production, community composition. And this allowed us to look at, so if you look across the natural world, you find more productive places have lower diversity. Theory suggests that it should be the opposite way. So here we're disarticulating nutrient supply rates from diversity. And what you find is that as you plant more and more species, you get more biomass. So relative yield is the production above a monoculture. So these at 1, this is the 1 species. And then as you go up to 16 species, you get more and more above ground biomass. And this is showing the years of the first 15 years of the experiment. And you can see that effect size increases dramatically over time. So this is another case where the experiment, every single year of the experiment, is producing a stronger and stronger effect of diversity. Now looping back, this is a diversity effect. Like I said, we have all these multiple changes occurring at once. So we may be losing biodiversity. We're also increasing nitrogen, increasing CO2, changing temperature, changing drought levels. So we have another sort of almost like hyperfactorial experiment at Cedar Creek where we're looking at the interaction of all of these with diversity. So this was put in 4 years later. So as soon as we learn, wow, look at these diversity effects, how are those going to interact with nitrogen supply since nitrogen reduces diversity? How are those going to interact with CO2? So if you look out in these fields, you can see these kind of weird space pod looking rings. So each of these rings has pipes all the way around, and these have CO2 being pumped out over them to create a dome of CO2 enrichment. So this is called a free air carbon enrichment experiment, a FACE experiment. DOE funded a bunch of these back in the late 1990s. This is the longest running open air CO2 enrichment experiment in the world. Actually, that diversity experiment is the longest running diversity experiment. Inside here, there's a bunch of smaller plots that are planted into different diversities of native prairie plants and exotic plants, with even some bare plots. Those, some receive nitrogen, some are control. And then in 2012, overlaid, Peter Reich and Sarah Hobby are running this experiment. They overlaid another experiment which adds drought and warming. So we have warming with lamps and also soil warming. So that's been running for 14 years. So I wanted to show you just a brief example of some of the results from this. So here's looking at effect size, just like in that previous graph. And you can see so many interesting things here. So eCO2, this is CO2 enrichment. The first decade, we get a really big effect of CO2, and then it just fades out over time. Nitrogen enrichment kind of builds up and stabilizes. And then these are the effects of the diversity treatment. So this is the difference between 1 to 4 species, 1 to 9, and 1 to 16 species. And like I showed earlier, you can see the diversity effects just keep getting bigger over time. And this is looking at total soil carbon, or total carbon budget in the ecosystem. So this is all focused on Cedar Creek. So I'm just going to briefly talk about ways to expand that knowledge more generally within an experimental framework. So this is a distributed experiment that I co-manage with Elizabeth Borer, who's another professor over in EEB. So we were really interested in these experiments, but you can do an experiment yourself at one place or maybe 3 places if you like to drive a lot. But really, science is seeking generality. So how do we have big observing networks and we have local experiments. So what can sit in between there? So we thought about, there's a lot of pretty simple experiments that can be done, and you can leverage some of the collaborative networks. So we've been working on building up this experiment with collaborators all around the world. And so this is combined 2 experiments. So one is looking at multiple nutrients. So it's nitrogen, phosphorus, potassium, and micronutrient addition, all in factorial combination. And then that's also crossed with herbivore fencing to look at sort of herbivore effects interacting with nutrient supply rates. And all of these nodes are all in grasslands all around the world, up in the Himalayas, in the Pampas, in Argentina, in Australia, across the US. And really, like I started out with, this is really exciting data, but more than that, it's a collaboration of hundreds of scientists around the world who are now interacting, sometimes coming to University of Minnesota, but not all of them, but collaborating and building really cool papers and knowledge. And a lot of these things are closely tied to some of the basic theory that's been developed at Cedar Creek. So for example, here's a graph showing the years of nutrient treatment and the nutrient effect on richness. And this now, instead of showing different fields at a site or different blocks, every one of these is a different grassland all around the world. And so you can see that this phenomena that we observed at Cedar Creek is actually very general. So almost every one of our grasslands, when we fertilize it, we start to lose species and that builds up over time. All right, so just to loop back, I wanted to kind of bring together some different ideas. We all work with data. We love bigger data, longer data, but have you just think about the role of people and collaborations in building science. Like, science is a social enterprise and emerges from people collaborating together. And that, you can think about data, but just automating data and spewing it out in the world isn't necessarily going to build our insights into how the world works. It's really individual people and groups of people thinking about critical questions. And I think it's an interesting point, too, with the rise of, it's like, oh, AI, machine learning, this is going to solve all our problems. But those things can't ask questions about what we don't know. When what really drives science is the frontiers of ignorance, right? It's the things that we don't know that are exciting, like what's the stuff. And that's what people do, is think about what we don't know. And I wanted to bring in this idea of experiments and how they can slot in and complement observational studies to lead to more mechanistic insights, and that there's really exciting ways to leverage collaborators and collaboration to get really big, exciting projects going across the world or across a state, and that this can happen through just building up collaborative networks. All right, then I wanted to just loop back with hopefully you're excited about Cedar Creek, but tired of hearing about grasslands and carbon. But so Cedar Creek is, like I said, it's been around a long time. We've got 5,600 acres. It's at a really interesting place continentally. So these are the big biomes of North America. So boreal forests. We have the deciduous forest to the east, the prairies. It's right on the edge, and so it switches between these different biomes depending on the land management. It's also got really exciting aquatic habitats. We've got really nice floating bogs. We've got really interesting lakes and streams. And we have a really big, it's got to go back to my notes here. Yeah, I always get excited about the research. But so we also have really nice facilities for working. So we've got 15 buildings on campus. We have housing for about 80 people. And like I said, it's only 45 minutes away. So it's also easy to drive up and do day trips, hold retreats, hold classes, bring your field classes out. You marched all your freshmen out through the nettles, right? I'm sure they loved it.
All of the very pretty freshman boys decided they wanted to wear shorts.
Yes, and they had very red legs afterwards.
They retreated.
Exactly.
And the girls laughed at them.
We also have, that's good. We also have really great outreach programs. So if you want to work on public engagement, so we bring in about 5,000 K-12 students every year who are out working in these experiments and working with scientists. We also have a really big public outreach program. So we bring in about 5,000 public visitors through talks and site visits. So if you're interested in community engagement, and we have education programs. So we have classes, field classes on site, all these kinds of things. And I'm just, so if you're interested in getting involved, I'm the director out there. Caitlin Burrell-Potter is associate director, and Mao Wei is one of our site scientists. You can shoot us an email. I'd also love to host anyone out there. It would be great to people, if there's enough interest, to have a visit in the spring when the water is flowing and there's good stuff to see. We'd love to take you around and show you some of these big experiments, also look at some of the aquatic habitats. I think it'd be great. So I'm happy to take questions now. Thank you for your time.
Just have a very naive, curious question. Is this an artificial meander or like a natural meander?
These are natural.
natural.
Yeah, and then the upper part of the creek has been ditched, so it's completely straight. But you can still see the old meanders. It's really cool. Like if you look on aerial photos, or actually the old topos, they're like little oxbow lakes now. And so rather than kind of recreating meanders, the plan is to just sort of put it back in its old channel.
Are there any like photos, time-dependent photos of the meanders over time?
Probably. You know, there are a lot of aerial photos, but I haven't gone back through and looked at them.
Thanks.
Hello, thank you. In terms of the experiments where you're seeing changes in richness, do you ever get species that say were in the seed bank and recover or spread from other plots? How do you account for like an increase in richness compared to like the original? Does that happen?
Yeah, for sure. And there's multiple ways that can happen, and it happens at kind of different time scales. So, you know, you can get recruitment of species out of the seed bank, so that can happen in a year. And then there's dispersal. So if you look at some of these old fields, some of them are more isolated, and the richness will remain low for much longer because some of the species just take a long time to disperse into there. And we're doing a lot more with looking at dispersal. So we've got a couple people, so Lauren Sullivan at Michigan State and Adam Clark, who's at Graz in Austria. So they took a whole bunch of the species and put them in a wind tunnel with their seeds, you know, and basically measured their dispersal kernels, and then used that to model the time it takes for them to show up in fields. And you can get this, it really predicts pretty well how long it takes for the species to show up. So that's a great question, yeah.
Thank you.
Very cool talk, thank you.
No, thank you.
I was curious, early on in the talk, you mentioned that y'all are doing things like experimentally grazing bison and stuff out there.
Right.
Is that like, what area are they covering? Do y'all have them kind of restricted to a certain area or over most of the size of the preserve?
Yeah, so right now we're in a pause, but so what we did, I don't have a map of the reserve, but the southern half is divided up into burn units.
Cool.
And then we created a 200-acre sort of long, skinny rectangle that spans across the burn frequencies,
Right.
and then put in exclosures inside there. So one of the ideas, so savanna systems require fire to maintain them, but it's a bit of a dilemma because the more you burn, it also prevents the seedlings from recruiting. And so one of the ideas was that the bison can graze. They don't really like the woody vegetation, so they're more like cows, so they're eating grass. Their grazing can reduce the fire intensity and create a more heterogeneous fire landscape, and that can allow some of the oak seedlings to come up. And it seems like it's been working. Cool. So it's really looking at the interaction between the grazers and the fire frequency in terms of restoration.
Interesting. And how long have they been there for?
I think we had them in for about 4 or 5 years, and then they've been off last summer. We had a commercial bison producer, which worked for quite a while. Like he would just bring them in and let them go and then pick them up in the fall. It just ended up, whatever. It wasn't economical for him, and right now we're sort of restructuring. And I think something that'll be more interesting sort of scientifically for how we source the bison, so we're in a little pause, but yeah.
Cool, thank you.
Yeah. They'll be back.
Thank you. I love hearing about nitrogen, so that was fun. I have a question about thinking about managing a living lab space, because it seems like you have scientists that come to observe the site itself over time, and then also scientists who construct long-term experiments. And how do you think about the space in terms of its ability to evolve naturally over time, and then disrupting it with maybe setting up a bunch of plots or whatever inputs a scientist might bring. And I guess maybe the size is just big enough where that isn't a concern. But if you had a tiny living lab space like our outdoor stream lab,
Right.
I'm curious how you would think about it as both a place to observe over time as this constructed ecosystem evolves, and also a place where scientists can come and meddle for their own, to answer their own questions.
Yeah, no, that's a great question, and that's, so we have a bunch of different parts of that. So one is it's almost kind of like a zoning question. So the vast majority of Cedar Creek looks like this, and then you saw a couple places that look like a farm field. We have some areas that we designate out for sort of more, intrusive, manipulative kind of experiments. So we just have zones where we don't really do anything, like it's kind of observational only. At the next scale up, there's this question, like what is we were talking about this over lunch, like what is a natural landscape? Like this landscape, if we didn't do anything, it would just turn into forest. And so, we do burn it, we burn the prairies, and so there's a land management component of, which makes it artificial in a way, but also maintains some of the diversity and tries to keep natural processes like fire, which would normally have been coming through, but aren't now because it's, whatever, we're in a rural landscape where people aren't that excited to, let the fires go through. But I think also the other part is like we have these plots where they can be quite small and people want to do a lot of sampling. And so some of these that's like really high value sort of real estate. And so we have, some of these experiments have been around for, over 40 years. And so we have the plot like mapped out. And so it's like, well, I don't know, can we really take one more soil core? Where is it going to go? And so it gets into some sort of, maybe quantum physics, sort of like you can't, whenever you observe something, you change it, right? And so going in and measuring things alters it to some degree. So that's not really an answer, but maybe a reflection. Yeah. I try to put more photos like this because people love that, like that photo of the biodiversity experiment, but it makes people think that the whole place is just like an ag field. But like most of it's just left alone. And, we do management like fire, invasive species control, but a lot of the stuff out here is just observational. So it's not really having much of an impact. Yeah.
So most of it was a great talk. Thank you very much. Most of the talk was about doing terrestrial ecology with all these test plots. Is there an aquatic part of the facility that actually that you could do anything similar to that, where you have control for nutrients or blah, 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 look at diversity, but from an aquatic point of view, or not?
No, not for experimental work at this time. It could be, like setting up mesocosms or something, but the aquatic work we've done has been more in the natural systems, like, doing monitoring in the lakes, doing, bag experiments in the lake, and then doing monitoring in the creek. I think the creek restoration is setting up some really cool things because the creek runs from a natural sort of headwaters through a ag landscape into Cedar Creek where it's ditched, and then down and out again. And, that's going to get reworked. So I think there's some quasi-experimental opportunity with the restoration and also kind of moving from between different land uses that could fall into that realm. But I don't think anyone's taken advantage of it, which is why I'm here.
It seems like it maybe could be done. You could partition that creek that we look at here
Right.
into test zones. So you could
Exactly.
look for what kinds of inhabitants you could.
Yeah, absolutely.
There's things
And it kind of happens on its own too, because we have a kind of disconnected chunk which is kind of the headwaters, which is natural. It goes through this sort of ag landscape. It goes back on the reserve, crosses a road, back on the reserve, and back into an ag landscape. So you could almost just pull out those different reaches and see what was happening. You've probably thought about this more from being up there, but.
Hi, thanks for your talk. I was kind of curious about the specific experiment where you showed that carbon storage stayed relatively high even after you stopped fertilizing, and like for many years, and what some reasons for that might be, and if you would eventually expect it to decline, and like how long? I guess maybe that is something to be determined.
Yeah, no, that's a great question, and this gets into kind of how carbon is stored. And so we're more and more looking at kind of the carbon fractions and what parts are associated with the minerals versus just being in the organic matter. we have a collaborator who's been doing more work with carbon isotopes, so you can actually age the carbon as well. And the carbon, turns over more slowly as you go deeper in the soil, but also the mineral-associated carbon turns over much more slowly, which gets into these kind of microbial, like detailed processes. When the microbes get in on the minerals and are like storing carbon right on it, it can be there for centuries. But it's a little bit unknown, like that's an area that's just kind of being worked on. And so that's kind of like a broad thing. Like it seems like the carbon is sticking around. Another, as a follow-up on a related experiment, instead of stopping fertilizing, we took half the plots and burned them every year. And I'm working on a paper on that one. And that one came out in the great way of science, like completely different. Like I was like, how much carbon and nitrogen are we losing by annual burning for 30 years? And it turns out we're gaining a huge amount of carbon. And that by burning, you're actually increasing the plant growth, and that the carbon gains from increased production are far outweighing the losses through volatilization, which I completely did not expect. So there's more soil carbon, like 30 years of annual burning in a grassland led to more soil carbon. I went back over and over, it's like, have I got my code backwards or what?
Yeah, kind of as a follow-up question to that, it seems like in addition to all these great science mechanisms and questions you can address with these experiments, you also are uncovering new insights into how these fields could be managed for carbon sequestration.
Yeah.
So what do you tell farmers that have fallow farmlands in this case? Right, like have you been getting into that conversation?
I can tell you're in the urban LTR because you're talking to social scientists.
What kinds of conversation do you have in terms of how you can get other people to manage their fields, or even thinking about policy around incentivizing farmers to manage their land for carbon sequestration?
Yeah, one of my collaborators on these projects, Andrew McDougall's at University of Guelph, so near Toronto, and he works a lot with the Canadian wheat growers, and they're very excited about this because they're working on policy, but trying to have realistic policy, right? Because they get this well, take your land out of production forever and we'll pay you this much. And they're like, well, we need to make a living too. And there's a lot of interesting areas where, so one thing is that as we get better and better data, we can find the area, even small scale areas in fields that never really pay off, right? They're just bad. I mean, you drive through Iowa or western Minnesota, you can see these cornfields, and then it's wow, that's a terrible spot for corn. And so, that those areas can just come out because they're costing the farmer money. But then we're also working on some modeling projects where, what are return intervals for tilling where you're still gaining? Maybe every tilling round you're losing 20% of the carbon. So how long do you have to leave it fallow to still be getting carbon gains? And there's also kind of two parts of that curve. So there's the carbon acquisition phase, so where you're storing more and more, and then there's kind of an asymptotic part where it's storage, and both of those have to be worked into policy, and so there's a lot of things going on, but I kind of Andrew is really great, and the farmers are super excited about it because I feel like it's helping them make the case to the policymakers that this is really valuable, but there needs to be some nuances in how the policy is written so it's realistic and allows them to make a living.
You're going to tell them to burn their fields every
No, I know, exactly. Well, there's some nuance, it's oh, we should just fertilize. I was talking to an NPR reporter, it's no. Don't write about how everybody should fertilize their fields to make the environment better. This is not the story. Yeah, no, great question. And I'm really glad to work with him because he's right out in the fields with the farmers, and they're super excited about it. So,
I think we have time for maybe one last question. isn't the last one, then I'll go for the last one. So what I noticed when you were showing over 40 years, the changes in overall above ground biomass,
Yeah.
that was going up about 10%. And I was curious whether that is related to the time scale essentially to reform the carbon stocks in the soils.
Right, that's another great one where, I mean, that's what I think is probably happening, and you know, that as you get more carbon, you get more cation exchange capacity, you get more water holding capacity, you're more effective at storing, because it's also I didn't show this, but we're also storing the nitrogen at the same level. And so then the nitrogen's retained more effectively, all the micronutrients. And so there is this positive feedback as, you know, as plants grow, they're making the soil better in a way. So yeah, that's exactly right. And you see that with like fertilization effects having increasing effects on biomass that keep growing. You know, why is that? Well, it's probably because the soil's getting better, right?
Eric, thank you for coming all the way from the St. Paul campus.
Oh, yes.
Beyond the Minneapolis
like the
campus.
best parking in the whole university, so it was easy.
Yeah.
And maybe we can loop back. I mean, if anybody you want to send me a note and think about going out to Cedar Creek, or maybe we can loop back and kind of try to make a field trip if people are interested and see some of these cool habitats. I'll listen while people who know about aquatic stuff talk about the streams. You can see some pitcher plants. I mean, who doesn't love a carnivorous plant, you know?
All right.
Well, thank you so much. It's been great.